Good morning to everybody. Would like to thank you, the organizing committee, for giving me the opportunity to present here this work. I'm going to talk about the growth hormone receptor in xenotransplantation and the hypothetical effect on complement activation and coagulation. One of the main issues in xenotransplantation coming out from the last observations are that the organ, when it's transplanted from pig to baboon, for example, or into a human, may still have the potential to further grow. And earlier observation from kidney xenotransplantation and also heterotopic pig to baboon xenotransplantation showed at the end of the experiment an overgrowth of the graft. And this overgrowth of the graft can be due to two reasons. One, of course, is the rejection mechanisms, which are fluid and cells that collect inside the graft and in edema formation. And the other one may be due to the physiological growth. So the organs still continue to grow, maybe because there is an upregulation of the growth hormone receptor in pig organs. This is an example of the heterotopic xenoheart transplantation. And the donor heart at the beginning of the experiment was smaller compared to the baboon recipient heart. And after the experiment, after several months, this is the result. So we can see an increase in the size and also the organ increase in gain. Another interesting finding is that Laron syndrome people, so people affected by the growth hormone receptor mutation, seem to be protected against cardiovascular diseases, type 2 diabetes, and cancer. Let's have a look at the growth hormone receptor IGF-1 axis, the pathway. So when you have a release of the growth hormone in circulation, you have that it will bind to the specific growth hormone receptor on cells, on the organs, and it will lead to the activation of the intracellular pathway, adjuncts that pathway, and this will lead to the secretion of insulin-like growth factor 1, which will bind to its own receptor, IGF-1R, that will lead at the end after transcriptional regulation to the growth. IGF-1 is reported in literature that is also important for endothelial cells homeostasis, so for the vasodilation, for example. So we were asking, does the absence of the growth hormone receptor have an effect on complement and also on coagulation? This is mainly, this is an in vitro experiment, but in vivo experiments are already planned. As you can see from the picture, pig lacking the growth hormone receptor are already available, and we are planning to transplant organs from this pig into a growth hormone receptor positive pig to see whether the organ continue to grow or not. So first of all, we made a modification on our porcine aortic endothelial cells. These cells already are lacking of the alpha-gal antigen. And as you can see here, after CRISPR-Cas technology, we induced a mutation on both alleles of these cells for the growth hormone receptor. And after a characterization, these cells showed that they still maintain the endothelial phenotype. So we have CD31 expression, V cadherin, von Willebrand factor, and even heparan sulfate proteoglycans. We checked also the expression of the growth hormone receptor, both in wild-type cells and also in growth hormone receptor knockout cells. And this antibody is against the intracellular part domains of this receptor. And as you can see here, the cells still produce the growth hormone receptor, but you can see that it's mostly perinuclear localization in the growth hormone receptor knockout cells, while in the wild-type cells, you can see that there is also membrane localization of this receptor. We carried out cell ELISA experiments by treating the cells with normal human serum for 1 hour, and we checked immunoglobulin deposition, IgM and IgG. As you can see from the graph, here we have the absorbance, and we didn't see any differences between GAL knockout, growth hormone receptor knockout cells, and the only GAL knockout cells in immunoglobulin deposition. this deposition is lower compared to the wild type, of course, because of the alpha-gal knockout. Then we checked also the effects on complement deposition, C4BC and C3BC deposition, and this is the same case, so we didn't find any differences between the two cell lines. Then we carried out an in vitro model that allow us to check the clotting time of the whole human blood. So with this model, we are able to increase the surface that will be in contact with the human blood by culturing the cells on microcarrier beads. And this is an example of immunofluorescence staining because before doing the experiment, we need to be sure that the microbeads are fully covered with cells. This is a CD31 staining because the microbeads itself are really procoagulant. After culturing the cells on these microbeads, we incubate the cells with non-anticoagulated freshly drawn human blood, and after incubation 37° and gentle mixing, we measure the time until clotting. So as you can see here, in this experiment, we used gal knockout human CD46 human thrombomodulus cells, and we compared the results with the growth hormone receptor knockout cells. As you can see, even in the clotting time, there are no differences between the two phenotypes. And this is a really high clotting time because mainly of the human thrombomodeling. We are talking about 3 hours of clotting time, and when we have wild-type cells, clotting time is about 50 minutes. So these cells continue to delay the clotting time, but not further more than the human thrombomodulus CD46 and gal knockout cells. So in conclusion, the growth hormone receptor deficiency on porcine endothelial cells with the gal knockout background retained the endothelial phenotype, and we didn't see any differences in immunoglobulin binding, complement deposition, and the anticoagulant properties. And the used in vitro test did not reveal a significant benefit of the deletion of the growth hormone receptor. Of course, maybe we need a more integrated system to see the real effect of this growth hormone receptor. And in vivo experiments may provide a better understanding on this, on long-term effect of this deletion. As an outlook, we would like to check whether the growth hormone receptor IGF-1 axis is really disrupted by these mutations. And so we need to do functional evaluation studies. For example, IGF-1 measurements will be a good idea to measure this secretion on supernatant before and after growth hormone treatment. And testing, of course, other clones coming out from this CRISPR-Cas modification. I would like to acknowledge my supervisor, Robert Reben, and all the other collaborators. Thanks for your attention.
Thank you very much. David?
Yeah, thank you very much. That was very interesting. If I get it right, do these pigs that have growth hormone receptor knockout, do they grow at all?
After 4 weeks, they seem smaller compared to the other pigs. So you can see a growth retardation after 4 weeks.
But say at a year, have they grown since 4 weeks?
Sorry, I didn't get.
Well, if you follow them for a year, do they continue growing or they stop?
I don't know, because so far our collaborators that are producing these pigs didn't reach 1 year of survival of these pigs. So.
Because as you know, we've found very considerable growth of the kidney, as Peter Friend's group did many years ago, particularly during the first 4 weeks, and then it sort of levels out. But I think with the kidney it's not a problem, but with an infant, say, with a heart transplant, it would be a problem, because if the heart's growing inside the chest, it will be a problem. But my question is, do we know if that heart or kidney will ever grow quickly, because that's what but will it ever grow as the child grows? Do we know that? Is that known?
I don't know. I can hypothesize that by the deletion of the growth hormone receptor, we still have in the organ the IGF-1 receptor. So when you transplant this organ lacking this growth hormone receptor, you have that in the other organism you have circulating IGF levels, normal levels, that maybe they can be higher in a child because of the growing phase, and maybe the organ can grow with the child. I don't know actually, but
Thank you.
Thank you.
Yeah, we have to be very careful when we are measuring these hearts during long-term survival, because what we have seen, and also Dr. Cooper mentioned, that once the rejection sets in, I mean, the heart sometimes grow. And what you showed there was a rejected heart, and that definitely grows, to a larger size. Mm-hmm. But what we have shown is, like, for a long period of time, I mean, if you suppress rejection, the growth is not at that much. So my question is that, I mean, is it possible to differentiate between, the heart that's undergoing rejection versus where the rejection is suppressed? So see if the growth hormone plays any role in there.
Yeah, that's a really interesting question. I don't know if we can measure this during the xenotransplantation. But yeah, it's a good question because, of course, rejection is the main mechanism of this overgrowing of the organ. But we are just hypothesize this.
Well, we've had 3 kidneys now. We clearly did not have rejection for at least more than 6 months. And yet, during the first month, they doubled or tripled in size. So I'm quite convinced that it's not just rejection that's causing this growth.
But it does stabilize, right? I mean, so did the heart. They grew for a few months and then they stopped growing. So,
yeah,
I mean, same as with the
But
kidney.
I think in that first month, there still seems to be some pig growth hormone around that makes them grow. But I don't think it's at all related to rejection.
Thank you.
Thank you very much.
Thank you.